introduction. Thanks very much to the Smithsonian for inviting me here today. You mentioned the fact that I got my PhD at Duke, and I just want to mention my pleasure of seeing my PhD advisor coming to the talk unexpectedly as a surprise. So I just want to wave to everyone. Yanis, this is Yanis Antonovic, an outstanding PhD advisor. So I'm sure many of you would like to talk with him afterwards. So today I'm going to talk to you about the promise of using digital herbarium specimens in large-scale climate research. This is a topic that I've been thinking about with my colleagues, Natalie Ivanicki-Alstrand, Matt Austin, Zoe Panchin, Christine Romerman, and Abe Miller-Rushing. So for many years, people have recognized that phenology, the timing of events, particularly the flowering time of plants, would be a very strong indicator of the effects of climate change. So we might expect other effects of climate change on plants, such as the changing distribution of plants or the changing abundance of plants. But of all of these, it's the changing of phenology, which has proved to be the most sensitive indicator of plants of the effects of climate change on plants, but even of all biological systems. It's particularly the timing of plant flowering in the spring, which has proved to be a very sensitive indicator of climate change. And I'm sure, again, all of us know that climate change is being driven by the increasing use of fossil fuels, the burning of coal, oil, and natural gas, and the increasing levels of carbon dioxide and other greenhouse gases in the environment. So for the past 20 years, my students and I and colleagues have been investigating the effects of climate change on plants. And one particular resource which has proved to be surprisingly effective at identifying the effects of climate change is herbarium specimens. And I think we've all been talking about herbarium specimens. I'm not sure if anyone's given the definition of it yet, but herbarium specimens are dried, flattened specimens of plants that have labels on them describing the date of collection, where they were collected, and the collector. And the question that we started asking about 20 years ago was, can you use these herbarium specimens in climate change research? And the answer is obviously yes, you can use these. And we started using these about 23 years ago at the Arnold Arboretum in Jamaica Plain in the city of Boston, Massachusetts, the Arnold Arboretum of Harvard University. And at the Arnold Arboretum, we started monitoring the collection for the timing of flowering, and then we found that they actually had a herbarium specimen collection of more than 100,000 herbarium specimens collected on the grounds of the Arnold Arboretum. So we were able to take our observations of when plants were flowering and then matching them with when plants had flowered in the past using these herbarium specimens. And again, herbarium specimens are very suited for this because herbarium specimens are often collected when plants are in full flower. I also just want to mention that this is one of our photographs. And one of the things that our research group does is we take lots of really beautiful photographs, posed photographs that we can use to tell the stories of climate change. So we actually have about 500 versions of this one particular photograph here. And this is just showing an extremely beautiful specimen of rhododendron vasei in full flower and combining that with herbarium specimens of this exact same plant which were collected in previous decades to tell the story of climate change. And what you can do is you can take herbarium specimens. Each one of these dots here represents a herbarium specimen, and this is shown in relationship to when that plant flowered in comparison to a reference year of 2003. And so what you can see on this graph here is that over time, from the 1880s to the present time, that plants have tended to flower earlier now than in the past. So in the past, plants tended to flower later than the reference year, and then in more recent year, they tended to flower earlier than the reference year. So over time, we see that the plants are flowering earlier. So this is an indicator of the effects of climate change. But instead of looking at the data over time, we can take the exact same herbarium specimens for hundreds of different plant species, and we can see whether they are flowering earlier in warm years. And what we find, as you can see in this graph, is that in warm years, let me get my pointer here, so that in warm years, plants are flowering earlier than the reference year, and in cold years, they're flowering later. So that the reason that plants are flowering earlier over time is not because time is changing, but it's getting warmer. And I still remember in 2003 when we made this figure here. So instead of looking at changes over time, I asked the students I was working with if they could just plot the data in relationship to spring temperature, and they got this figure like this, and I couldn't believe it. I thought maybe we were doing something wrong. But since then, we've produced results like this over and over again, and other people have. So this is the first result demonstrating that you can use herbarium specimens to look at the effects of climate change, and the results are so striking. And now this result has been repeated by many research groups around the world, and it clearly demonstrates the value of using herbarium specimens in climate change research. So you can also use herbarium specimens to look at other phenological phenomena of plants. And one of these, for example, is to look at the effects of climate change on when plants leaf out. So you can use herbarium specimens like this sugar maple here. And when the leaves are coming out in the spring, the leaves are smaller in size, they're often a little bit wrinkled, and they're pale in color. So you can use these to tell when plants have leafed out. And again, often, particularly for trees, they often flower and leaf out at the same time in the spring. So herbarium specimens often represent a good record of when plants leaf out. And the results are the same for trees as well. So trees have been leafing out earlier over time across the eastern United States, and they leaf out earlier in warm years than in cold years. So until about 8 years ago, when myself and other people around the United States and other countries have been using herbarium specimens in climate change research, you often had to go from herbarium to herbarium to get enough data, or you had to work at a herbarium like the Smithsonian that has a lot of specimens. But it was often a road trip to do this kind of research. And so most of these kind of herbarium studies were local, like at the Arnold Arboretum. And similarly, most phenological studies were often very local, people working at specific sites or at networks of specific sites. And so one example of a local field site, which was mentioned, is that we were building on the observations made by Henry David Thoreau in the 1850s. And what we were able to show is that plants are now flowering, observations made by Henry David Thoreau in the 1850s, and we were able to show that plants are now flowering earlier than they did in Thoreau's time, and that they flower and the trees leaf out earlier in warmer years. But we wondered whether this local study done in Concord, Massachusetts, one small town in New England, whether this really was happening, whether this phenomenon was really happening across a larger area. Also, what we found in Concord was the trees were more responsive to climate change than the wildflowers. And we wanted to see whether this phenomenon of trees being more responsive, more sensitive to climate change was really happening over a larger geographical area. And it turns out that this was really a good time to be thinking on these larger scales, because as people have mentioned, herbarium specimens started to be digitized really actively around 15 years ago. And this is a graph just showing how many herbarium specimens have been digitized. And you can see that it really took off starting around 2010. And since 2010, about 6 million herbarium specimens have been digitized per year in addition. So we're increasing the number of herbarium specimens, about 6 million specimens per year. But if you look at where those specimens are from, about 20 million of those specimens are from the tropics. So most herbarium specimens are from the temperate areas, relatively small number from the Arctic. So we're getting a huge increase in the number of digitized herbarium specimens. So these are specimens which are photographed digitally and are made available online, but most of them are still from the temperate area. And so starting about 4 years ago, myself and various colleagues were interested if this phenomenon that we saw in Concord, Massachusetts, of species being responsive to climate change and trees being more responsive than wildflowers, whether this was true across Eastern North America and whether this was true in Concord. And so what we did is we used digital herbarium specimens to get information about when trees were leafing out and when wildflowers were flowering for each of these continents. And for each continent, we focused on 6 common wildflower species and 6 common tree species. And so just for Eastern North America here, so this represents about 2,000 specimens. The pluses represent a herbarium specimen of a tree. The circles represent a herbarium specimen of a wildflower. So that's about 2,000 specimens across there. We know the date and the location, and so we can match that with PRISM weather data to know what the spring temperature was for that place and that year. So we know what the temperature conditions are. And so we have all the phenological data for trees and wildflowers going back 130 years at these particular places and the phenology. One thing I want to point out here is that there's a lot of data there for Eastern United States, but also it's not randomly distributed. See, it tends to be really concentrated on the East Coast, certain other cities or mountain areas, but mostly you're kind of along the East Coast. So it's a non-random distribution of specimens, but we can deal with that in our analysis. We also got equivalent data from Europe and East Asia by working with colleagues from those countries, and we all combined in terms of comparing trees and wildflowers. And this is what our results look like. So from Asia, Europe, and North America, one of the things which is really striking about this is if we look at the spring temperature and when these plants were flowering and when the trees were leafing out, you can see that in all of these continents that we can see the phenomenon that trees and wildflowers are responding to spring temperature. When it's warmer, they flower and leaf out earlier, and when it's a cold spring, they leaf out and flower later. But we have these very striking differences between continents. So first, if we look at Eastern North America, that trees are more sensitive to climate change than the wildflowers. So what we saw in Concord is really true across Eastern North America. In Europe, the wildflowers are flowering much earlier than the trees are leafing out. And then the other thing that I want to point out, which is really striking, if you look at these R-square values, you can see that the R-square values for Eastern North America are much stronger. So trees in Eastern North America are responding much more strongly to temperature than on these other continents. And so this suggests a whole series of questions that might be worth further investigation, all coming about through using herbarium specimens. I also want to share with you some other examples from other researchers that I think really represent the most exciting thing that you can now do with these digitized herbarium specimens. So this is an incredible study that was published last year by colleagues led by Ramirez Parada. And in this study, they were interested in finding out whether phenotypic plasticity, so the ability of plants to adjust their phenology, was more significant than evolutionary genetic adaptation of plants to their local climate. And so they used 1 million herbarium specimens representing 1,600 species and looking at the flowering time of plants all across North America to address whether species in one location tended to have very variable flowering times, which suggested phenotypic plasticity, or they tended to be very fixed in one place for their genetics, or fixed in one place for their phenology, but varying from place to place in their phenology. So this is really an incredible study using an enormous number of species, all with digitized herbarium specimens. Their results are very complicated, very challenging to understand, but their major conclusions were based on this figure here, which is also hard to understand. I'm not going to explain this figure, but only to say that their conclusion was that phenotypic plasticity was more important than genetic adaptation, that plasticity, phenotypic plasticity, so variability in response to temperature at the local scale, was more important in the spring and the autumn, and that plasticity was more important in the southern United States and other regions. So again, they're suggesting a whole series of very interesting questions which further detailed genetic and ecological studies could contribute to based on herbarium specimens. I want to mention another study which is using herbarium specimens to address a very novel ecological question. So there was a study led by Wano and colleagues who were interested in investigating the phenomenon of hysteranthi, of flowering before leafing out in American plums. And so they looked at 12 species of plums in North America, and they looked at all these plums all across North America for when they flowered in relationship to leafing out. So here we have a plum on the left, illustrating the phenomenon of hysteranthi, of flowering before leafing out. And what you can see here is that this is a species which has this very dramatic visual display. And in this research, the group was interested to know whether it was characteristics of the flower, the size of the flowers, the number of flowers per inflorescence, or ecological characteristics which were more important in explaining hysteranthi, the phenomenon of flowering before leafing out. And this figure on the right shows their main conclusion. So what we have on the x-axis is an index of drought tolerance, and on the y-axis is a measure of hysteranthi, of how much the species tends to flower before it leaves out. And you can see there's a negative correlation. And what that negative correlation, each one of these dots here represents a species, and each one of these dots represents about 200 specimens, digital specimens of that species. What the results tell us is that the species which flower well before leafing out tend to be found in the driest places, and the species which leaf out and flower at about the same time are the species which are found in the moister places. So it suggests an ecological explanation for hysteranthi based on herbarium specimens and kind of aridity data. I want to mention a study showing the connection between climate change and reproductive biology, again, using these digitized herbarium specimens. A study published very recently by Shi and colleagues, in this case using poplars. So poplars have male and female inflorescences. And in this article by Shi and colleagues, they wanted to find out if there was any difference in the flowering time of the male and the female inflorescences on these species of poplars, populus species. They used 9 species of populus. They had 13,000 digitized specimens, so roughly about 1,300 specimens per species. And again, they wanted to know if there was any difference in the when the male and the female inflorescences were on the plants. And this is an example of some of their data. So in this figure here, you have data for 9 species of poplars, and it's saying, based on these digital herbarium specimens, when are the flowers open of these male and female inflorescences. The blue are the blue and gray represent the male flowers, and the red bars represents the female flowers for each one of these 9 species. And you can see for each one of these species here, the male flowers are open before the female flowers. But there's another striking result of theirs, which they also looked at the flowering time in relationship to the temperature, and they found that for each of these species that the male flowers were more sensitive to temperature than the female flowers. And what this means is that as the temperatures get warmer, the differences between the female and the male flowers are going to get greater, and at some point in the future, they might even have completely separate flowering times, in which case the male flowers are not going to be able to fertilize the female flowers, and there's going to be potentially a reproductive failure in the case of these poplars. So a very kind of unusual reproductive, evolutionary, ecological result, all coming from herbarium specimens, which can then suggest further field studies. I also want to mention how herbarium specimens can be used in urban ecology. So people are very concerned with how cities, and particularly the warming of cities called the urban heat island effect, might be affecting ecology, how it might be affecting the timing of flowering and other phenomena in cities. And there was this quite amazing study by Park and colleagues published recently, where all across North America, Eastern North America, they looked at counties and looked at the herbarium specimens from particular counties. They looked at 71,000 digitized herbarium specimens for more than 200 plant species. So that's roughly about 300 or 400 herbarium specimens for each species. And they were interested in seeing how responsive plants were to the warming of cities in those particular areas, or the density of human populations. And what they found was that there was an earlier flowering time of cities in comparison to adjacent rural areas, so an urban effect on phenology, but it was primarily in cooler and cities that were found in cooler or warmer areas, shown in the kind of the dark red areas of this figure here. And they also found that there was a delayed flowering, but particularly in places that in urban areas that had wetter springs. So this tells us that there was an urban effect on phenology, but it was tied to the local climate of that place, or the particular climate of temperature and rainfall in that particular place. So all coming from these digitized herbarium specimens. I also want to mention just how herbarium specimens, digitized herbarium specimens, can be used to inform new ways forward in terms of doing phenology research. I want to mention this study done from Denmark using 3 species done by Ivanicki Ahlstrand and colleagues on horse chestnuts and a few other species. And what was really neat about this study by Ahlstrand was that she and her colleagues were looking at whether you could combine herbarium specimens with first flowering observations or potentially with iNaturalist photographs. And what you can see here is in the upper graph is that herbarium specimens, that there weren't that many herbarium specimens that she was able to find from Denmark for these different species of onions, elderberries, and horse chestnuts, and that the herbarium specimens were found from only a limited number of places in Denmark. There was also a citizen science program to get people to make first observations, which is shown in the middle here. The problem with first flowering observations is that they're not exactly comparable to herbarium specimens, which are mostly specimens collected when plants are in peak flower. And then the lower figure here shows observations or photographs taken by people who are trying to identify specimens using the iNaturalist platform. So people uploading photographs and then asking the iNaturalist what they are. And so you can see on the bottom here, these iNaturalist photographs, this relatively recent citizen science network has generated a huge amount of data. People are very enthusiastic about taking photographs with their smartphones and then getting a system to identify it. So there's an enormous number of relatively recent photographs taken with the iNaturalist platform. And they also cover very densely the area of Denmark. And what's also very exciting about iNaturalist photographs is not only the lot of them, but their numbers are increasing every year quite dramatically. And so what Ahlstrand concluded was that it's possible to combine herbarium specimens, which are specimens collected when plants are in full flower, with photographs taken again when plants are in full flower, and you can really combine them to do to gain better insights into the effects of climate change on plants. So I also want to mention that when one is doing this kind of research with herbarium specimens, it's very important to be aware of the biases and potential problems of using herbarium specimens. If you're not aware of the potential biases and problems of herbarium specimens, you might get a very interesting result, but it might be just an artifact of the biases and the problems of using these specimens. So for example, if you're not aware of the geographical biases of collections, you could come up with an erroneous result. Or when these specimens are being digitized, it might be possible that there are mistakes which are made in the digitization process or the reading of the labels, which could give you erroneous results. There are sometimes ethical issues. For example, if you're using specimens without getting the proper permission, or you're not giving acknowledgement to the herbaria where your material is coming from. There's also problems of herbarium specimens that many geographical areas are underrepresented, particularly areas in the tropics. Just as kind of an example of the biases, people should look at the paper by Daru. There's this amazing paper by Daru published about 8 years ago, where he and his colleagues describe a lot of the problems of biases in herbarium specimens. But just to take one example here is if you look at New England here, the areas shown in blue are underrepresented, and the areas shown in red are just excessively collected. And you can see within New England, there's this huge variation in the density of collections depending on where you are in the region. Also, biases in collection. So this is a figure here showing the number of specimens collected by different collectors in New England, represented by digitized specimens in New England. And you can see there's this guy, Les Meyerhoff, and he's collected something like 20,000 specimens. And then you have something like 20,000 collectors that have each collected one specimen. So depending on what Les Meyerhoff liked to collect and where he liked to collect, it really biases the kind of data that you have. So I just want to mention a few other things here, that there are important future directions. So there are many exciting topics in ecology and evolution that you can use these specimens for. this area, it's a very exciting area because specimens are continuing to be digitized. So we can already do a lot with digitized herbarium specimens, but more are continuing to be collected in coming years. Also, the quality of digitized herbarium is getting better as we get better at gaining more information using artificial intelligence during the digitization process. As artificial intelligence gets better at reading the labels and making the label information available on the specimens. And also the standardization of digitized herbarium specimens is increasing so we can combine digitized specimens over a larger geographical area. And so in conclusion, what does this all mean? So I hope I've convinced you that digitized herbarium specimens allow us to do exciting ecological, evolutionary, genetic, conservation studies over a much larger range than was possible before, and also using a much larger number of herbarium specimens. It's possible to take local studies like the study of Thoreau in Concord, Massachusetts, and really expand this to a larger geographical area. It's important to be aware of biases in this kind of research, because if you don't consider the biases, you might get an interesting result, which is just an artifact of the biases. These new resources, and particularly using artificial intelligence to get more characters off of these specimens and appreciate the labels better, allows us potentially to ask new questions. So it's a very exciting time to be a botanist. We have to be concerned with the declining number of taxonomists. We have to be concerned with the threats to herbaria. But at the same time, it's a very exciting time to be a botanist investigating questions of phenology and climate change, and especially using digitized herbarium specimens. Thank you very much for your attention.
Yeah, hey, Richard. So we have a couple questions online. The first question is from Joseph, who asks, do you consider that the timing of collection may have changed at different times?
But the timing of collection?
The timing of the collection may have changed at different times. So when are people collecting their specimens?
You know, it's possible. I mean, one could imagine that people went on botanical expeditions. So again, I know a lot about New England. So in New England, there used to be large groups of people that used to go out with the New England Botanical Club, collecting huge number of specimens. A lot of specimens were often collected by botany classes going out, and that kind of pattern is changing. So I think that's one thing changing. Another example, which is a really dramatic change, is that people in the past, I think, avoided collecting non-native species. And then particularly people like Les Meyerhoff really focus on invasive species. So there have been these changing patterns of collections, which really might have affected the kind of results that people get. That's something we don't know that much about, but that's potentially a very interesting topic.
Great. Thank you. And then the second question is from Elaine, who asks, in the populous species, some of the species show that male and female flowers do not show overlap. Does this indicate that these species are rarer or have smaller distributions?
Say again, that they...
So does this indicate that the ones that show that the male and female flowers don't overlap, are they rarer or do they have small distributions?
Well, I think they all show some overlap. I mean, you have some overlap between the males and females, otherwise they'd be complete reproductive failure. So I think that, you know, I think that these are mostly widespread species, which is why we have a lot of specimens of them. But they still all have overlap, so don't worry about the populous species quite yet. But worry for the future.
That I don't have any emphasis. I don't know all these popular species. Some of them have quite extensive clonal reproduction. If there's any variation, or that the authors of that article.
Yeah, the question is whether these species are clonal and whether that might have affected the pattern. That I'm not sure of. One thing which I do want to mention, since this is a topic that I'm especially interested of the phenology of male and females, is that the female flowers might stay longer on the plants because they sort of are persistent, because they will be there eventually developing into the fruit, whereas the male flowers might be briefer. And this might explain part of the pattern that collectors might have had a bias toward collecting the male flowers because they were more conspicuous, and then had a bias toward the female flowers because they were more persistent.
Great talk, and thank you. I have two questions that might be only one. So you see these differences between trees and shrubs in the way they respond to flowering over time. Do you think this difference of trees is because these other plants can migrate altitudinally going up, just to avoid maybe the warmer weather, or is just because they are just different? And this also comes back to, do you think, I know you probably don't have enough data, but in the tropics, do you think we're going to find similar patterns? Or do you think as it happens before in evolutionary times, that plants are going to go up when climate change or the weather becomes hotter?
Okay, not sure I got it, but I mean, what we think is happening is that perhaps the trees are, because they're in a warm environment, sort of higher up, and they're in the air, they're more responsive to climate change because they're just responding to the air temperature, whereas the wildflowers are in the ground. And so the surrounding soil has to warm up, and that might make them a little bit less responsive to variation in temperature. But I also want to mention, we didn't find this phenomenon in Europe and in Asia. And also there was another group that was investigating the same phenomenon using citizen science data from Eastern North America, and they didn't get the same results we did. So we've actually trying to reconcile their results and our results.
So do you think there's something about perennial and annual plants there too,
in the case
or
of
not?
these wildflowers, they're all perennial wildflowers. None of them are annual wildflowers.
Okay, thank you.
Okay.
Hi, thank you, Dr. Primack. I don't know if this is on. Can you hear me okay? Okay. So I just wanted to ask, you said that the phenology of flowering had changed, but I was wondering if, I was wondering if you also saw a difference in the amount of time that a tree remained in flower over the years. The length of time. Yeah, like the length of time it was blooming.
So the question is there any difference over the time that plants remain in flower? That I'm not sure of. I would have to kind of think about that. People probably have investigated this. It just sort of escapes me at the moment. But I think in general, I mean, as conditions get warmer, then the flowers tend to stay less time on the plants, because within a season, if you have a warm weather, then the flowers last just a few days. And if it's very cool weather, especially cool weather, cloudy weather without much rain, the flowers can last weeks. So we'd have to kind of think about that. Thanks very much.
Thank you.